Assalamu alaikum, dear students. Welcome again to the online microbiology classes. Today we are going to discuss a lecture number 2, and in this case, today's title is Branches of Microbiology. We will discuss a detailed description of various branches of microbiology. So let's start. This slide is going to present the list of various branches of microbiology. These include pharmaceutical microbiology, medical microbiology, industrial microbiology, food microbiology, soil microbiology, and plant microbiology. Literally, as the name indicates, they are self-explanatory, but we will have to discuss because they are very much important. Pharmaceutical microbiology and medical microbiology is of paramount importance. However, industrial microbiology, food microbiology, soil microbiology, and plant microbiology, they are also equally important. First one is pharmaceutical microbiology. It is that branch of microbiology which has special bearing on pharmacy in all its aspects. This ranges from manufacture and quality control of pharmaceutical products in general to an understanding of mode of action of antibiotics. As the name indicates, pharmaceutical microbiology, it means that microbiology which has special effect on the pharmacy. And you know that in pharmacy profession, we deal with the manufacturing of the pharmaceutical products. Now, this manufacturing of pharmaceutical products occur in the pharmaceutical firms or pharmaceutical companies. So during manufacturing process, different quality control tests are performed. Now, at the end of manufacturing, also the quality control tests are performed. These quality control tests are important because they give us clue that our manufactured product is accurate and up to mark. Now, you know that there are different types of the pharmaceutical products and there are different types of dosage form. For example, tablets, for example, syrup, for example, creams and ointments. As that dosage form is changed, now the nature of the quality control test also changed. So it means that there are different quality control tests for the tablets, for the capsules, for the syrups, for the elixirs, for the creams, for the ointments. Now these tests are performed during the manufacturing process as well as when the manufacturing is complete. And what is the significance of these quality control tests? They give us clue that our manufactured products, they are up to mark. Now generally the microbiology which affect on the pharmacy, and since pharmacy deals with the manufacturing of pharmaceutical products, there are different kinds of the pharmaceutical products. So all the pharmaceutical products generally they are affected by the pharmaceutical microbiology. But you know that in case of microbiology, we deals with the microbes, and in case of microbes, one most important microorganism is the bacteria. So in order to kill the bacteria, antibiotics are used. Now these antibiotics, they are also manufactured in the pharmaceutical firms or pharmaceutical companies. And now this is important to know about the mechanism of action of these antibiotics. You know that these bacteria for which antibiotic act as a killing bomb. Now the structure of the antibiotic, if you have in your mind the structure of antibiotic and bacteria in your mind, then you can easily understand the mechanism of action of the antibiotics. You know that outermost layer of the bacteria is cell wall. Now, so there are certain antibiotics which affect the cell wall, which inhibit the cell wall. As a result, there will be no cell wall, so the bacteria is also killed. However, inside the body of the bacteria, proteins are also present which are responsible for protein synthesis. So there are certain antibiotics which affect the protein synthesis. As a result, proteins are not synthesized, and as a result, bacteria are killed. You know that bacteria contain DNA. So there are certain antibiotics which kill the DNA. As a result, bacteria is killed. So we can say that in order to understand the mechanism of action of antibiotic, first of all, you must know the vital organs of the bacteria. You must know the structure of the bacteria. You must know the different parts of the body of the bacteria, because when you know about these parts, then you will be in the better position to understand the mechanism of action of the antibiotics. Here, this is the structure of bacterial cell. Now this is outermost layer is called cell wall. Inside the cell wall there is plasma membrane or the cell membrane, and then there is the cytoplasm. And in the cytoplasm you say that there is DNA present, and then there is transcription and translation. And you know that this transcription and translation, they are actually protein synthesizing intermediate processes. That first of all there is transcription. In this case, messenger RNA is formed, then there is translation in which the protein synthesis take place, and then the replication, then other similar genetic materials, they are synthesized. So first of all, there are certain antibiotics which inhibit the cell wall. Outermost layer of the bacteria is cell wall. So when they inhibit the cell wall, it means that this cell wall is not present later on. When there is no cell wall, it means bacteria cannot survive, so bacteria kill. So inhibition of the cell wall synthesis. So there are certain antibiotics, for example, penicillin, cephalosporin, bacitracin, and vancomycin. These are the names of the antibiotics. When we take these antibiotics, first of all, they destroy the cell wall of the bacteria. As a result, bacteria is killed and we are safe from the infection. So these are the antibiotics, and this is the mechanism of action, that these groups, penicillin, cephalosporin, bacitracin, and vancomycin, they inhibit the cell wall, and as a result, bacteria is killed. Secondly, there are certain antibiotics, for example, chloramphenicol, erythromycin, tetracycline, and streptomycin. They inhibit protein synthesis. They have no effect on the cell wall, but inside the cytoplasm, they inhibit this transcription and translation process. As a result, there is no messenger RNA and there is no protein synthesis. When the proteins are inhibited, ultimately the bacteria is also killed. So these antibiotics, chloramphenicol, erythromycin, tetracycline, and streptomycin, they inhibit this transcription and translation. As a result, they inhibit the protein synthesis. This is the second mechanism of action. Third, you know, then this is the DNA. There are certain antibiotics which inhibit the nucleic acid replication and transcription. Now, as a result, there is no there is DNA synthesis inhibition. For example, quinolones and rifampicin. These are the antibiotics which act on the DNA. As a result, inhibit its replication and transcription, and as a result, no DNA is present. As a result, bacteria is killed. This is the third mechanism of action. Fourth one, injury to plasma membrane. For example, polymyxin B, this antibiotic has no effect on the cell wall, has no effect on the protein synthesis, has no effect on the DNA, but they affect the plasma membrane. They injure the cell membrane. As a result, they kill the bacteria. So this is another mechanism of action. And last one is inhibition of the synthesis of essential metabolites. For example, sulfone amides or sulfonylamide and trimethoprim. They inhibit the synthesis of essential metabolites. As a result, they inhibit the bacterial growth. So it is quite easy that the pharmaceutical microbiology, which is having special focus on the pharmacy, and you know that in the pharmacy, we deal with the manufacture and quality control of pharmaceutical products in general. But since in case of pharmaceutical microbiology, we have to deal with the antibiotics, so it is only possible that we can understand the mechanism of action of these antibiotics if we know about the structure of the bacterial cell, because certain